Hi, my name is Andrea,
I'm Andrew.
And I'm Matthew.
and we're going to be giving you guys a lecture today about the chloroplast and its development.
All right, so to start, just some quick announcements for you guys. Final exam is on Thursday, March 20th. I'm sure you guys are all preparing very hard. Just make sure to study. I'm sure you guys are if you're here. You all seem locked in. But if you guys need any help, we will have office hours specifically about Chloroplast Development on Wednesday, March 19th from 1 to 3 in the morning. So if you guys are interested in that, go for it. So here are some of our learning objectives for this lecture. This is what we want you guys to understand, which we want you guys to learn, and hopefully our lecture encapsulates most of this stuff for you guys.
So we have a clicker question for you guys, and that is, where did the chloroplast originate from? So we have option A, mutation in the plant cells, option B, a fungi that formed a symbiotic relationship with the plant, C, it originated as a cyanobacterial symbiote, or D, who knows? Does anybody know the answer? Well, let's see. It originated as a cyanobacterial symbiote. And let's get into the chloroplast development.
All right, so just a quick overview before we begin. Development is obviously different in dark light versus direct light as it is in most cases with plants. In developed leaves, chloroplasts are further propagated by fission, similar to bacteria. And something we really want to highlight is epigeal germination versus hypogeal germination. In epigeal germination, the cotyledons first serve as storage organs before throwing off the seed shell and becoming photosynthetic above ground. And in hypogeal germination, the cotyledons stay below ground and the epicotyl grows while the hypocotyl remains the same length. And this is just important for us to highlight because most of the plants and systems we'll be talking about in this lecture are going to be hypogeal, which is more common in plants. Here we just have a diagram kind of going over what I said. You guys can see the epicotyl and the hypogeal germination slowly growing above the ground. Yeah, just a quick example for you guys.
Okay, so here we have a simple visual representation of plastid development. As you can see in this figure here, when the plant is underground, it's obviously in the dark, so the plastid will develop into an etioplast. However, once the plant reaches sunlight, the plastid will differentiate into a chloroplast, which we all know is important for photosynthesis. So in the dark, we have a pathway that's very similar to the light signaling pathway that we learned about a few lectures ago. So in the dark, phytochrome cannot enter the nucleus. When this happens, both phytochrome and cryptochromes, aka Cry, are unable to suppress COP1 and SPA1. COP1 suppresses high 5, and PIFS suppresses chloroplasts and photosynthesis-associated nuclear genes. And when this happens, no chloroplast biogenesis and division can occur.
So when we are in direct light, the phi and the cryptochromes, the cry, are actually activated and they can accumulate in the nucleus. So when this happens, as we can see, phi inhibits the PIFs and the COP1, whereas cry inhibits the SPA1. So with this complex being inhibited, we now allow for the activation of the high 5 complex. So this allows for the accumulation to contribute to the activation of the CP and PHANGS, which allows chloroplast biogenesis and the chloroplast division. So the question of the day that we have for you guys today is, why do plants hate math? And we'll get back to this at the end of the lecture. So here's a quick overview about the processes involved in chloroplast development. We have all of the steps here, and we're going to get into them one by one for you.
So one of the first steps in chloroplast development is the protein import and processing. Proteins enter plastids via TOCs and TICs and are directed to their destination via HSP90 and TOC159. It was discovered through experiments that chloroplast biogenesis is severely impaired when TOC 159 is knocked out. Imported proteins become integrated into the thylakoid membranes and lumens, which contributes to their overall structural development. Next, we have gene transcription and translation. In this step, sigma factors including SIG2 and SIG-S, along with nuclear encoded RNA polymerases like RPOT, are needed to initiate chloroplast gene transcription. Nuclear encoded proteins called PPRs are also critical for RNA processing, splicing, editing, stability, maturation, and translation in the chloroplast.
So next we're gonna get into protein folding and degradation within the chloroplasts, or to form the chloroplast, more to speak. Chloroplast protein folding is mediated by chaperones such as HSP70, which is expressed at high levels in both seedlings and adult plants. And interestingly, this gene is actually highly conserved for protein folding in all organisms, not just plants. Redundant genes further along the pathway, like CpHSC71 and CpHSC72, harbor ATPase activity, which is essential for the HSP70 chaperone activity. So as you guys can see here, like I said before, CpHSC71 is essential for development. Specifically looking at the right image, we see an example of wild-type plants, growing in a normal environment, in a normal way. And on the right, we have a plant with the mutated CpHSC71 gene, which can result in issues with leaf formation, chloroplast development, and ultimately the death of the plant. So continuing further with protein folding degradation, degradation is extremely important for chloroplast development. The multi-subunit CLPPR proteinase complex actually plays a central role in degrading proteins that is required for chloroplast protein homeostasis. Loss of this can either result in embryo or seedling death. And it's just important because certain mutations can result in deaths on their own. So without this type of regulation, you can see serious issues with the plant development. So moving further down the path, we see thylakoid formation and pigment biosynthesis beginning. The thylakoids are formed mainly by mono and digalactosyldeacylglycerols that are either produced within the chloroplast or at the endoplasmic reticulum and then move to the chloroplast. During this formation, you'll also start to see the photosynthetic complexes formed, which is where you'll start to see pigment formation, as well as excited electrons finally begin to enter the cell, and you'll start to see photosynthetic processes begin.
So getting into the next step, we see retrograde signaling. So most of the proteins needed for the chloroplast function are actually encoded in the nucleus. This necessitates a tight regulation between the chloroplast functional status and nuclear gene transcription. So the signaling from the chloroplast is involved in regulating the nuclear transcription, which we see here in this step. So onto the chloroplast division. Chloroplast division is mediated by tubin-like proteins and diamine proteins that form concentric rings within and outside the chloroplast envelope. Some of these are called, the tubin-like proteins are called FTSZ, and the diamine proteins are called ARC5, which stands for the accumulation and replication of the chloroplast. So the diamine protein divides symmetrically, maintaining proper distribution in plant cells. And a typical plant cell contains between 100 and 300 chloroplasts. So we have one last clicker question for you guys, and that is, what is the role of chloroplast in the cell? So we have A, allows for nitrate sulfur assimilation, B, biosynthesis of some amino acids, fatty acids, pigments, nucleotides, and several plant hormones, C, sensing environmental stimuli and stressors, D, photosynthesis, or E, all of the above. So what do we think it is? So it's actually E, all of the above. And then back to our question of the day, why do plants hate math? It's because it gives them square roots. Thank you guys for coming to our lecture today, and we really appreciate it and hope you guys learned about chloroplast division. Bye.